Good afternoon, everyone. We'll be starting with the class, and today we'll discuss the concepts that are discussed in week 4. So, let's move on to question number 1. I hope my screen is visible. So, the first question is match the genes with the class of lower organ identity genes which they belong to. So here in the column 1 the name of the genes, and in the column 2 this is the class of the genes. So can you guess what can be the answer? So these are the classes of genes A, B, C and E, and here as we can see in class A there are two genes, that is AP1 and AP2. So we have AP1 and AP2 in class A. In class B we have AP3 and P1. So in class B we have AP3 and P1, and in class C we have AG gene, and in class E we have SEP genes. So for A it's 1 and 6. So 1 will be 1 and 2 and 6, and for B it's 1 and 4, and for class C it's AG, that's 3, and for class E it's SEP1, that is SIP5. So the correct option will be B, because it joins all the correct options. So this is the explanation for the question, and as we have discussed, AP1 and AP2 belongs to class A genes, and AP3 and P1 belongs to class B genes, and AG genes belong to C class, and E class corresponds to SEP genes. And these genes are important in developing the different floral organs of the plant. And here we can see that how different organs, different floral organs are developed using the combination of different genes. Okay, so any doubts in this question? Anyone?
Sir, these genes are only activated only on the meristematic regions only, no, sir?
Sorry, can you repeat?
Is that these genes?
So we are moving to the next question. So the question states a mutation in a model dicot plant resulted into having altered floral organs as carpel, stamen, stamen, and then carpel. So what, according to the ABC model of flowering, which causes the mutation in which kind of which class of mutation is there in the plant? So it can be A class gene mutation, B class, C class, or A or B double mutant. Can anyone guess?
B class gene mutants.
B class gene mutant. Any other guesses? So the correct answer is option A class gene mutants. How we're going to see here. So like we have just discussed that if like only sepals will be present, then A and E genes will be present, and for petals B, A and E genes are important, and for stamens development B, C and E genes are important, and for carpel it's C and E. And here we have like if A class genes are mutated, then it will form carpel, that is C and D, then stamens, that is B, C and D, then again stamens and carpel. So this is what the question is asked. So this carpel, stamen and carpel. Now, what about if B class genes are mutated? So in that case, as we know that for sepals only A and E are important. So it will be sepal, then carpel. This arrangement will be there when B class gene will be mutated. And now if C class genes will be mutated, then in that case, so A and E will form sepals, then BAE will form petals, then again petal, and then it will be sepals, and it will be in n numbers. Okay, and when E class genes are mutated, so what happens in E class? E class is important for development of all the floral organs. So in that case, it will be leafy growth. So nothing will be differentiated in that case. Okay, everyone understood?
Yes, sir. I have one doubt, sir. Actually, eukaryotic genes are monocystronic only, no, sir. Then, what is this representation of all these five kinds of mixed genes has been represented here, sir? Since it
Okay.
is seems to be a polycystronic.
So this is not on the basis of monocystronic or polycystronic. These genes helps in the development of the particular floral organ. So for example, if we talk about sepals, so what happens is, as you know, that leafy is the one that moves towards the floral meristem. So when leafy is there, it activates the gene class A genes first. So for example, it will activate AP1 first. Okay, then when these genes are activated, the products that they are forming, they activate the B class genes. Okay, so it's a pathway that they act upon. So it's not like that these genes are arranged in a manner that the genes are monocystronic or polycystronic. It's the way they are acting during the development process.
Yes, sir. Yes, thank you, sir.
Okay, so any more doubts? Or shall I explain it again if anyone have not understood? If not, then we are moving to the next question. So this is the next question. Which of the following statements is false about pin proteins? So the options are: they can be asymmetrically localized in the membrane, they can always be placed symmetrically in the membrane, they are auxin efflux carriers, and the last one is: the Arabidopsis pin mutant lacks defined shoot apical meristem and it is pin shaped. Can anyone guess the answer?
Option yes, sir.
Option A.
Yes.
Okay. Any more guesses?
Sorry, sir. They have told false. Option C, sir.
C. Okay. Any more guesses? Hmm. So first to solve this question, we must know where the pin proteins are involved in the signaling pathway. So pin proteins are mainly involved in auxin efflux. So this is correct. This is not wrong. Okay. And what happens is when suppose this is the cell, auxin enters from this protein, that is IAA, and then it has to form a polar transport. So the it will go out from the cell using the pin proteins that is localized on the other side of the membrane. So what this arrangement tells us that it is not symmetrically located in the membrane, it's asymmetrically located in the membrane. Okay, because there is always a polar transport for oxygen. And so option A is correct and option B is false. Now how does this pin name came? And because when the scientists were developing the mutants using this protein, so they saw that the SAM region of the pin mutants in Arabidopsis is not clear and it is not well defined. So and it's pin shaped. That's why the name of the protein came to be pin. Okay, so the correct answer is option B because it's not symmetrically located. And here is the explanation. So that's what I was telling, that IAA ox is the influx transporter of oxygen and pin is the efflux exporter. So it will be localized only on the side where the cell wants to send the oxygen out from the cell. So it will not be symmetrically located on the membrane. And when these pin mutants were created in Arabidopsis, they don't have a well-defined shoot apical meristem, and they were pin-shaped, like shown in this figure. So these are pin-shaped. That's why the name of the protein is pin. Okay. So anyone have any doubts in this? Okay, then we'll move on to the next question. So these are the regulatory proteins in column A and we have to match them with column B. So the proteins are pin protein, fabulosa, and Wuschel. So, what can be the correct answer in this?
Option A, sir.
Option A. Okay. Any more guesses? Okay, so just now we studied that pin proteins are involved in auxin efflux carriers. So A will be 2. Okay, then auxin 1 or like AUX1 is an influx carrier of auxin. So that will be 3. So B will be 3. And fabulosa is involved in leaf polarity. Okay, because it will be expressed in the adaxial way. So it is 1 and C is 1, and Wuschel is mainly important for SAM maintenance. So it will be 4. So yes, the answer is A. And like what I said just now, that fabulosa is involved in the polarity of the organs, and it is involved in adaxial polarity, and and Wuschel is important for leaf maintenance. Sorry, SAM maintenance. Okay, so anyone has any doubt in this question? If not, then we'll. Sorry.
It's a clear, sir.
Hmm. Okay. So we'll move to the next question. So, which of the following organelle covers the plasmodesmata in plant cells? So, the options are ribosome, nucleus, rough endoplasmic reticulum, or smooth endoplasmic reticulum. Can anyone take a guess? So the answer is smooth endoplasmic reticulum, and this is the basic structure of plasmodesmata and how it looks like. So this is the cell 2 cytoplasm, and this below one is the cell 1 cytoplasm. And what happens is this is the smooth endoplasmic reticulum that connects both the cells, and this is the plasma membrane and the cell wall. So this is the membrane structure of the plant cell, and here it's a callose deposition that uh, maintains the size exclusion limit of the plasmodesmata. Okay, and what like the proteins that are formed in the reticulum can pass through this directly, or the molecules that are small enough that can pass through these spaces. Okay, so anyone has any doubt in this?
So generally, cell contains endoplasmic reticulum has been surrounded near the nucleus, no, sir.
Hmm.
But whereas in plants, it has been arranged in different manner, or it has been arranged in the same manner like as we generally saw near the nucleus, sir.
Like it will be like that endoplasmic reticulum is of two types, that is rough and smooth. Yeah. So the rough endoplasmic reticulum is arranged near the nucleus only. Like if you see that this is the nucleus, and when the RNA comes out from the cell, so it undergoes protein synthesis. So the rough endoplasmic reticulum has ribosomes, which helps in protein synthesis of the proteins that are destined to cell surface or outside the cell. Okay, so rough ER is located near to the nucleus in every cell, being it a plant cell or animal cell.
Yes, sir.
Okay, and if we are talking about the smooth endoplasmic reticulum, it can be near or away from the nucleus, because it has no role in protein synthesis, but it has role in lipid synthesis and drug detoxification, like those. So it's not necessary for smooth endoplasmic reticulum to be present near the nucleus.
Yes, sir. So in plants, it has been added near the cell also, sir.
Uh, Sorry,
So
I
like in
missed this
plants, it
one.
has been added in a cell wall
Hmm.
region, sir.
So in this case, what is like what is there is that smooth endoplasmic reticulum is extended between the two cells, that will help the cells to carry out the communication or the exchange of all the materials that happens. Okay, so it is available in the cell also, as well as at the cell wall also.
Okay, sir. Thank you, sir.
Okay. Any more doubts? If not, then we are moving to the next question. So, leaf development displays polarity, and the leaf side towards the meristem is called adaxial side, and the side away from the meristem is called abaxial side. During normal leaf development, xylem is located towards the adaxial side and phloem towards the abaxial side. Okay, so xylem is located towards the adaxial side and phloem is located towards the abaxial side. So what would be the probable phenotype if the fabulosa gene has a gain of function mutant, given that fabulosa gene is specifically expressed towards the adaxial side? Okay, so you have a fabulosa gain of function mutation, and you have to find out what will be the phenotype of that plant. So these are the options. So either the xylem would be radial surrounding the phloem, or phloem would be radial surrounding the xylem. So can anybody guess?
So radial pattern of xylem sounds of phloem, sir.
So you are saying A.
Yes,
Okay,
sir.
any more guesses?
So, no, sir. If it's a mutant, so radial pattern of phloem sounds to xylems.
Okay, any more guesses? So the correct answer is option A only. And why? Because like in this, if you can see that the green colored is the xylem and the red colored is the phloem. Okay, so xylem is located on the adaxial side and phloem is located on the abaxial side. And as we know, fabulosa gene is on the adaxial side only. Okay, So in that case, if we have gain of function mutation in a fabulosa gene, so everything will be adaxially placed. Okay, so in that case, xylem would be radial to the phloem. Okay, but if it would be loss of function mutation, then everything would be abaxially placed. So in that case, phloem would be on the abaxial side, and phloem would be radial to the xylem, surrounding the xylem. So the correct answer is option A. Okay, any doubts in this question?
Sir, normal leaves, xylem surrounds the phloem only, no, sir. As xylem in adaxial side,
Hmm.
whereas in phloem in abaxial region.
Hmm.
So they have given that it has been mutated so that it will be in opposite manner only, no, sir.
No, actually mutations can be of two types. So one is the gain of function mutation and one is the loss of function mutation. So this is a gain of function mutation. That is if the like we have uh, the protein in extra amount, or the function of the protein is enhanced. In that case you can understand.
Okay, sir. Thank you, sir.
Okay, any more doubts? If not, then we'll move to the next question. So, which of the following statements is false about TFL1 terminal flower 1 mutant in Arabidopsis thaliana? So, will it be early flowering, late flowering, a single terminal flower, or reduced number of flower buds? So, can anybody take a guess?
So to reduce number of flower buds.
Okay. Any more guesses? Okay. So, the correct answer is option B, that is it will be late flowering, and as we know that TFL1 is the negative regulator of floral meristem, so it does not allow floral meristem to grow. So whenever there is a mutant Hmm?
Early flowering maybe the answers.
Uh, so that's what I'm explaining, that whenever there is TFL1 mutant, so as you can see in this picture itself, this is the wild type, this is the TFL1 mutant, as it is a negative regulator of flowering. So when this will be mutated, then this will there will be early flowering of the plant. Okay, so early flowering will be there because TFL1 is not there and it is a negative regulator of flowering. So early flowering will be there when is TFL1 is mutated, and when TFL1 is present, then it's late flowering. Okay, and we have a single terminal flower as we can see in this. So there are reduced number of floral buds, but there is a single terminal flower. Okay, so this is the thing that we observe in a TFL1 mutant or in Arabidopsis. Okay, so any doubts in this case?
So, if you mutated that TF1 genes, there must be early flowering only, no, sir?
Hmm.
Because TF1 is suppression of floral meristem. So, if we mutated, that means we can get flowering, no, sir?
Yeah, but we have to, you are right. You are absolutely right, but we have to figure out the false statement in the question.
Okay, sir.
Okay,
Thank you.
so these three options are correct and this one is false. So this is a false statement. Okay,
Okay, sir. Thank you, sir.
any more doubts? Okay, then we'll move on to the next question. So, in an experiment to decipher the role of gene X and gene Y during plant development, the mutants were analyzed for the phenotype. So, we are using two genes, gene X and gene Y, during plant development. So, the result showed that both X and Y single mutants are defective in lateral root development. So, both gene X and Y are single mutants. Okay, so in that case, one plant have only X gene mutated, and on the other plant it's Y gene mutated. Okay, and X is normal in this case, and Y is normal in this case. So, whereas in XY double mutant plants, the additive phenotype is observed. So, which of the following is best conclusion for the experiment? So, both X and Y genes have a role in lateral root development. X and Y genes function in different genetic pathways during lateral root development. C option is X and Y genes function in the same genetic pathways, or none of the above. Can anybody take a guess?
So, what does it you mentioned? They have shown the additive phenotype means what does it mean, sir?
Okay, so additive phenotype means that when sorry, so when there is mutation in both the genes. Okay, so the phenotype that is observed is, like, if we are studying the lateral root development, for example, so one pathway is toward from X and the other pathway is from Y. Okay, so when both the genes are mutated, then there will be very low amount of lateral root or no lateral root will be formed in that plant. Okay, otherwise, if X is only present, then some amount of lateral root will be observed, although diminished, and not as compared to the wild type. And similar is the case with the gene Y. Understood?
Yes, yes. Thank you, sir.
Okay, so So that is why in this question, if we are looking at this, so when X was mutated, then suppose we are studying the lateral root development, so this much amount of lateral root was visible, and similar was the case with Y. But when both the genes were mutated, so no lateral root was observed. Okay, so this was the additive effect of both the genes X and Y. So the option B states that both X and Y genes have a different genetic pathways during lateral root development. That is why when they both were mutated, they are showing a additive phenotype. Okay, so everybody understood?
So additive phenotype means the combination of both genes to contribute a different traits.
Yeah, it's basically the addition. So whenever there are two genes that are involved in a different pathway, but the same phenotype is observed, so that will give us the additive phenotype.
Okay, thank you, sir.
Okay, any more doubts?
So the additive phenotype may be over-regulatory or maybe a suppressed growth also, sir.
So additive phenotype depends upon the mutant that we are generating. Okay, so in this in this case it will be suppression, because here we are looking for lateral root development. But in some cases where there is we are mutating the repressors, so in that case it will be positive outcomes.
Yes, thank you, sir.
Okay. Any more doubts? If not, then we'll move to the next question. So, which of the following is a negative regulator of flowering locus T, that is FT, and suppressor of overexpression of constans 1, that is SOC1 genes during flower development? So, is it flowering locus C, that is FLC, fabulosa, terminal flower 1, and epithala AP3? So, can anybody guess?
Option yes, sir.
Okay, any more guesses? So the correct answer is option A. And fabulosa is, as you know, that it is involved in organ polarity, and TFL1 is involved in the maintenance of the SAM, and it inhibits flowering. And AP3 is the gene that is involved in floral organ development. So the correct answer is FLC, and here is the pathway that it is how it controls. So FT is flowering locus T, that is generated in leaves, and it transports to the apex of the plant via phloem transport. And as you can see that FLC inhibits both SOC1 and FT in both, uh, yeah, in leaf. So it suppresses its production only in leaf itself. Okay, so any doubts in this question?
In leaf regime, they have shown that there are various pathways that are suppressing the movement of FT, sir, towards after. Is that these ambient temperature, Gibberellins pathway are all suppressing the FT, whereas in photo period pathway, it will possibly regulating the FT to moving forward, sir. So, in, the majority here is the negative regulation of FTs has been shown here, sir. But whereas the photo period is only the thing which is possibly regulating the FTs, sir.
Yes.
So, how it has been overcoming the all negative regulations, sir?
Okay, so as you know, like in case of the photo period thing, so the timing of the daylight controls all the like all the phenomena that are happening in the plant. So what happens is when the photo period, like the plant in which it flowers appears, like if it's a long day plant, so the amount of sunlight it needs to flower is more. So what happens is at that time, this pathway gets activated, and this is during this pathway that all the other pathways are stopped. Okay, so in that case only this pathway is activated, and it activates the production of FT and its transport. But in other cases, like these things happens only when appropriate like in this case temperature and all those things are available at that point of time. So these pathways are not active all the time. Okay, so like in case of vernalization, it is the cold treatment. So when there is a cold treatment, then FLC inhibits FT and SOC1. Okay, so it doesn't take place all the time. It happens like that. So whenever that environmental conditions favors, then only these pathways get activated.
So, and yes, sir, it's clear, sir. And one more thing, sir. Here, the vernalization is inhibiting FLC. Um, then how it can be inhibit the FT? So here, vernalization is deactivating the FLC thing. Then
Yeah.
how it will be active to suppress the FLC?
Yeah, actually, like it's right that vernalization inhibits FLC. So that means that FLC will not work like inhibiting FT. So it is not like inhibiting FLC. Like it's different conditions. That what was I explaining.
Okay, sir. Thank you, sir.
Okay, any more doubts? If not, then we'll move to the next question. So, which of the following statements is false about the genetic pathways regulating flower development in plants? So, these are the options. Vernalization negatively regulates FLC and induces flowering. Vernalization positively regulates FLC and induces flowering. Photoperiod and gibberellic acid are positive regulators of flowering, and FT and SOC1 genes are positive regulators of flowering. So, can anyone guess? We have studied just now.
Option B, sir.
Okay. Any more guesses? So yes, it's option B. And as we studied just now, that vernalization is the negative regulator of FLC, and gibberellins are also negative regulator of SVP that inhibits FT. So these and as From this, we see that vernalization is a negative regulator of FLC that helps in inducing flowering. So it activates FT, and FT will move to the apex and it will help in flowering. And then photoperiod and gibberellic acid are also positive regulators of flowering because it inhibits the pathways that are inhibiting FT. And similar is this with the FT and SOC1 genes that are positive regulators of flowering. Okay, so any doubts in this question?
Sir, gibberellic acids are also negative regulators of flowering only, no, sir?
No, actually it depends because as we can see in this figure, the gibberellins are inhibiting SVP, and it inhibits like SVP inhibits FT. So gibberellins are like it is also context dependent, and so generally gibberellins are in support of flowering. That is our positive regulator of flowering. But in some cases, like when there is unfavorable conditions, so that in that case gibberellins are negative regulators of flowering.
So, whereas in apex region, gibberellic acid shown positive regulations, sir.
Yeah, in Apex also it shows positive regulation. It inhibits SVP here also, and
Yes.
it activates SOC and is a positive regulator of flowering.
Mm, okay, sir. Thank you, sir.
Mhm. Okay, so in this figure, uh, the blue ones that are shown are for activation of flowering, and the orange one or red ones that are shown are negative regulators of flowering. So everything that is displayed in orange or red, that is the negative regulator of flowering. Okay, so any more doubts in this question? If not, then we are moving to the next question. So, peptide signaling is an important regulator of development in plants. Which of the following are negative regulators of stomata development? So, EPF1, EPF2, stomagen, TFL1, or AP3? So, can anybody take a guess?
EPF1 and EPS2, sir.
Okay, any more guesses? So, the answer is option A. And as you can see in this figure that stomagen is the positive regulator of stomata development, and the gel is also a negative regulator of stomatal development, and they both EPF1 and EPF2 are also negative regulator. So how they work? They work via the ERF receptors. So these gel and EPF1 and 2, they bind to ERFs that are their receptors, and they inhibit stomatal production. But when stomagen is there, it binds to through TMM, that is its receptor, and it inhibits EPF1 or EPF2, and it helps in stomatal production. So any doubts in this question? If not, then we are moving to the next question. So, with reference to the leaf development, what is the correct sequence in the formation of leaf? So, is it blade initiation, then intercalary growth, then distal growth, then founder cell recruitment, intercalary growth, distal growth, then founder cell recruitment, and then blade initiation, founder cell recruitment, blade initiation, intercalary growth, or distal growth? So the correct answer is option D. And here it's the explanation of the question. So first what happens is SAM, that is the stem apical meristem. So it initiates the founder cell recruitment, that is the cells are destined that will form the leaf. Then what happens is there is distal growth, like if this is the primordia, so there is distal growth of the cells. Then there is blade initiation, that is first the blade of the leaf is formed, and then after that there is intercalary growth of the leaf. So these are the steps that are followed in the leaf development. Okay, so any doubts in this question? If not, then we'll move to the next question. So the size of Arabidopsis shoot apical meristem decreases in X mutant, increases in Y mutant, and is absent in Z mutant. So what are these X, Y, and Z genes respectively? So option A is Wuschel, clavata 1 or shoot meristemless, or clavata 1, Wuschel or shoot meristemless, shoot meristemless, Wuschel or clavata 1, or Wuschel, shoot meristemless or clavata 1. So can anybody take a guess?
Option yes.
Okay, any more doubts? Oh, any more uh, answers? So this was the last session's question also. So in this case, if you remember, Bushel gene is involved in the maintenance of the shoot apical meristem. So when this will be mutated, its size will go down and its size will decrease. Clavata 1 is the negative regulator of Bush. Okay, so when clavata 1 is decreased or is mutant, then the size of shoot apical meristem will increase. And shoot meristemless is the gene. In if this is mutated, then there will be no shoot apical meristem in that case. So the option is option A, and this is the explanation. So as you can see that this is the wild type of plant, and if Wuschel is mutated, so in that case you can see this is the central zone, and here the central zone is highly decreased because there is. And then if we consider that this is clavata 1 mutation, so in this case as you can see that the central zone is increased in this case because is it is the negative regulator of Wuschel. And in the third case, you cannot see any clear differentiation of the meristem. Hence meristem is not present in shoot meristemless mutants. Okay, so these are the different mutants that can be present in the SAM, and these genes are involved in the development of SAM. Okay, so any doubts in this question?
So can you explain what is the role of shoot meristemless genes, sir?
So this genes is basically helps in the formation of shoot apical meristem during development. So if this gene is not present, then the meristem will not be formed in those plants.
Okay, sir. Thank you, sir.
So can anybody tell which Fox gene is involved in this Wush clavata pathway?
Wow. What do, sir?
Yes, VOX2 is involved in this pathway. So any more doubts in this question?
No, sir.
Okay, then we'll move on to the next question. So there are various types of primordia that takes part in shoot development. If one of the primordia is removed, then what will happen to the other leftover primordia? So option A is the other primordia will grow normally at the same position. The primordia will have diminished growth. The position of primordia will change with different growth patterns, or none of the above options is correct. Can anybody take a guess?
The position of primordia will change with different growth patterns, sir.
Okay, any more uh, assumptions? So yes, the option will be option C. That is, the position as well as the growth patterns of the primordia will change. And as we know that all the primordia that are present on the shoot, they basically are in a very coordinated way. They function in very coordinated way, and if any of these primordias, for example, if I1 is removed from here, and this is the normal positioning as well as hmm. Hello, am I audible? Hello, is am I audible? Hello everyone, am I audible?
Yes, sir.
Okay, sorry, my mic was disconnected, so I rejoined here.
No problem.
Is my screen visible?
Yes, sir.
So we were discussing there are various types of primordia that takes part in shoot development, and if one of the primordia is removed, then what will happen to the other left primordia? And the answer was C here. And I was explaining you about the uh, how it takes place. So these are the different primordia that are present during shoot development, and it develops into the normal plant. So these are different primordia: P1, P2, P3, I1 and I2. If, like, in for example, I1 is disrupted or removed from its original position, then all the primordias will be disrupted and their position will be disrupted and they will not function normally. So it will show disrupted growth as compared to the wild type, because all the normal position as well as the normal factors that are involved during the development of the shoe typical meristem or the different organ of the plant, it will be disrupted. Okay, so have anyone has any doubt in this? So in plants, what happens is everything happens in a coordinated way. So during development, if we have to move from one stage to another, the signaling factors involved and the primordia that will develop first, and that is a very sequential manner. That's why this happens. So if any of these primordia is disrupted or has some mutations, then it will affect all the primordia that are developing during that time. So it will have effect on the whole plant development. Okay. So we'll be moving to the next question. So the concentration of oxygen is high in dash while low in dash of shoot. So central zone or peripheral zone, central zone or boundary zone, peripheral zone or boundary zone, or peripheral zone or central zone. So can anybody take a guess?
Peripheral zone, central zone, sir.
Okay, any more guesses? So if you guys remember, like, so whenever there is a growth or development in the shoe typical meristem, so there is a central zone, and there is a central zone and there is a peripheral zone, so below that. So and there is some boundary zone that is between the primordia and the central zone. So what happens is, in this central zone there is very low concentration of oxygen, and in the peripheral zone there is high concentration of oxygen. Okay, so the answer is option D. Okay, and this is the explanation that I was giving. So this is the boundary zone, this is the peripheral zone, and this is the central zone. So in case of central zone, there is high amount of cytokinin present, not the oxygen. The oxygen levels are very less. And in case of the peripheral zone, that is the zone for differentiation. So here the oxygen amount is very high. Okay, so wherever there is increase in differentiation, then the oxygen amount will be high. Okay, so this is it. And do you have any doubts in this question?
Says that oxygen can be synthesized in central zone also, sir.
Yeah.
So there may be a possibility of high amount of concentration in also in central zone also, sir.
No, actually, there can be synthesis, but it's under question because it's not been known till now that whether auxin is synthesized in the central zone or not. But yes, some studies have found that there are genes present for auxin synthesis in central zone also. But as per the experiments in other studies, they have not found any the amount of auxin in central zone is less. So this is right now controversial because the more studies need to be performed for this study.
Okay, sir. Thank you, sir.
Okay. So let's move to the next question. So a scientist is working with Arabidopsis and he tagged cyclin with GFP. Okay, so this is a translation fusion that we talked about, and he found that the GFP expression was uniform at day 3, but the green color starts fading towards the base of the leaf as the development progresses. So this depicts that the differentiation of leaf is basipetal, acropetal, bidirectional, or there is no differentiation taking place. So can anybody take a guess?
In the basic petal direction, sir.
Okay, any more guesses? So, like in the question, we are tagging cyclin with GFP. So, cyclin is basically involved in cell division. Okay, so when there is high amount of cell division, there will be high amount of cyclin present. Okay, so when during day 3, they are saying that it shows uniform expression. So this is the part that they are observing, and they are observing full GFP expression in uniform in the part. But as the days progresses, the green color start faded and it comes towards the bottom of the part that they are observing. So sorry. So now what they are observing is the green part is shifting towards the base. So this means that the development is going. So the correct answer is A. And here is what I was explaining. So in our question we have tagged cyclin with GFP, but in this case they have used a beta glucuronidase reporter. So at day 4 they see a uniform expression, but as the days progresses the development also progresses and the division is only in the base of the leaf. Okay, and here it's very less, so the development is taking place in this direction, that is the basipetal direction. Okay, so anybody has any doubt in this question? If not, then we'll be moving to the next question. So you are working with Arabidopsis and you found that the whole leaf region is abaxialized. Okay, so as you know, there are two regions of the leaf. One is the adaxial part, and one is the abaxial part. So this region is abaxialized. What can be the appropriate reason, which is not the appropriate reason according to you? So it is a loss of function mutation of fabulosa, gain of function of YABI, gain of function of fabulosa, or both A or B. So can anybody take a guess?
Option C, sir.
Option which?
Gain of function of fabulosa, sir.
Fabulosa. Okay. Any more guesses? Okay. So first, in case of, like, for example, there is a gene, it functions. Okay. So when there is a mutation in this and it does not function. Okay. So this is a loss of function mutation. Okay. And if the same gene has something mutated or inserted in it such that it has constitutive expression and it functions normally without any inducers or something, then it's called as a gain of function mutation. Okay. So have you understood the difference between loss of function and gain of function mutation?
Yes, sir.
Hmm. So now, as we know that fabulosa is present towards the on which side fabulosa is expressed?
Adaxial, sir.
Adaxial. And yabi is present on the abaxial side. Okay. So when fabulosa has a loss of function mutation and yabi has a gain of function mutation, then the whole region will be abaxialized. Okay. So the answer would be option D. Okay. So here is the explanation. And like in case of protein pathways that we are talking about, so adaxial surfaces have all these homeodomain containing factors, that is fabulosa, fabulota, and revulota. So all these are present towards the adaxial side, and canadi and yabi are present towards the abaxial side. So, whenever there is gain of function mutation of YABI and loss of function mutation of fabulosa, so both the things will contribute towards the abaxialization of the leaf. Okay, and there are, so the next question is, you want to prepare a flower mutant in a plant and you do not need to develop the floral meristem. Which of the following genes will you mutate in order to get the desired phenotype? So the options are leafy, AP1, TLR1, or both A and B. So the correct answer would be both leafy and AP1. That is option D. Both A and B are correct. So the correct answer is option D, and the explanation is here. So as we know that TFL1 is inhibitor of the floral meristem, but it helps in maintenance of the shoot meristem. And both leafy and AP1 is inhibitor of TFL1 and they promote the development of floral meristem. So, if we want to develop a flower mutant in which floral meristem is not present, then leafy and AP1 can be mutated, and that will help us to to develop a flower that does not have a floral meristem. Okay, so in this figure also we can see that TFL1 is present at the branches that does not allow the plant to develop a floral meristem. That is, it is a negative regulator of floral meristem. And all those leafy as well as AP1, that is yellow or green, are present over the top that helps in the development of the floral meristem. Okay. So the next question is how to stop the simplistic way of cell-cell communication in plants. So the options are by increasing the amount of callose, by decreasing the amount of callose, by increasing the amount of glucose, and option D by increasing the amount of lipids. So as you all know that these are the cell walls, and via the cell walls, the endoplasmic reticulum passes, and this forms the plasmodesmata. And as we know that the size limit of this plasmodesmata is controlled by callose that are deposited in the cell wall. So, so by increasing the amount of callose, the size of the plasmodesmata can be decreased, and this will stop the simplistic way of cell-cell communication. While if we decrease the amount of callose, then the diameter of this plasmodesmata will increase, and this will allow the molecules to pass from one cell to another. So the correct answer would be option A by increasing the amount of callose. So this is what I explained just now. So this is the smooth endoplasmic reticulum forming the plasmodesmata, and this is the cell wall of the plant cell. And this gray thing is the callose that helps in managing the size exclusion limit of this plasmodesmata. So if we increase the amount of callose, this length will be decreased and the symplastic movement will be stopped. And if we decrease the amount of callose, then this will increase the size limit of this plasmodesmata, and the molecules can easily move from one side to another. So the correct answer is option A. So moving on to the next question. So scientists are studying the size restrictions of molecules that are transported by these plasmodesmata. They labeled 3 times GFP with a suspensor cell specific promoter. So this GFP, that is 3 times GFP, like this is GFP1, GFP2, and GFP3. So this is 3 times GFP. So this is under the control of a promoter that is suspensor cell specific, and that is found during the early stages only. So during the early stages, the GFP is expressed all over the embryo. So, considering this to be an embryo, so the GFP is expressed all over the embryo. And after the formation of early heart-shaped embryo, the GFP expression is only restricted to the suspensor cell, but not the heart shape of the embryo. What can be the possible explanation? So, after forming the heart-shaped embryo, so they found that the GFP is only expressed in the suspensor cell, but it is not moving to the heart shape of the embryo. So we have to analyze what can be the reason for this. So option A, the size of the plasmodesmata depends upon the stage of development. The size of plasmodesmata depends upon the amount of callose deposited. The size of plasmodesmata depends upon the accumulation of lignin, or both A and B. So if the protein is not moving to the upper part, that means the size of the plasmodesmata is decreased. Okay, because this 3 times GFP will be more in size as compared to a single GFP molecule. So the possible explanation can be either the size of this plasmodesmata is decreased, or the amount of callose that regulates the size of the plasmodesmata is increased. So option A and B can be the answer. So option D is the right answer. And this is what I just explained. So the scientists used suspensor cell specific promoter under only one XGFP. So here they saw that all the GFP molecules are distributed all over the embryo. But after some time, they found that this uh, GFP is only localized to the suspensor cell and not moving to the heart shape of the embryo. And secondly, they saw that if the this if we put two times GFP under this specific promoter, so it stays only in the heart shape and does not move to the suspensor cell. And this is also the same case where we have used three times GFP in the heart shape of the embryo. So now this is the possible hypothesis that they are applying, that during the early stages, there is a movement between the upper part of the embryo and the suspensor cell. So either this way or this way, both the uh, nutrients and particles move. But when the embryo develops and heart shape is formed, then the movement from heart shape to the suspensor cell is followed, but not to the upper side. And secondly, what we can see from this graph, that if there is only a particular size limit that is followed from heart shape to the suspensor cell. So here the size limit has been decreased because of the amount of callose deposited and the change in size of the plasmodesmata. So this is what the question was. Okay, so next we are moving to the next question. So you want to stop the root hair formation using CPC proteins. What can be the strategies that you can use? So either we can use a CPC protein knockout, or increase the amount of callose synthase, or use a miRNA based technique to decrease the expression of CPC. So yes, the correct answer is all of the above. And here is the possible explanation. So, as we know that CPC is involved only in the cells that helps in the formation of root hairs. So, in the roots, there are two types of cells present. So, one will be H, that is the root hair forming, and the second one will be the non-root hair forming. So, CPC protein is involved in the formation is involved in the formation of root hairs, and but it is produced in the cells that do not produce root hairs. So it is produced in these cells and it is transported to the other cells, to the neighboring cells via plasmodesmata. So if the if we have to stop root hair synthesis, then we can develop a knockout with the CPC protein, or we can increase the amount of callose synthesis that will decrease the movement between these two cells. So that will not allow the CPC protein to move to this cell and it will not function as a in the formation of root hairs. Or like we have discussed, if in the miRNA system, that it can help decrease the expression of a particular mRNA or protein. So yes, it can also be used in decreasing the amount of CPC proteins. So all three options are correct, and either of the way we can use to develop CPC protein knockout, and that will inhibit the formation of root hairs. And this is one of the technique how we can increase the amount of callose in synthesis by callose synthase. So callose synthase is the enzyme that helps in the formation of callose, and increase in callose synthesis will help in deposition in the cell wall that will regulate the size of the plasmodesmata as we discussed before. And beta 3 glucanase is the degrading enzyme of callose, and that will also help in regulating the size of plasmodesmata. Okay, so now we'll move on to the next question. So the next question that's sieve elements. So companion cells are the living component of phloem, and sieve elements are the non-living component of the phloem because they do not have a nucleus. So all the activities of the sieve elements are controlled by the companion cells. So what we so they are in a constant uh, support of each other, and they are connected with plasmodesmata highly. So the question states you are expressing a protein coupled with GFP in the companion cells under the control of a XVE promoter. So XVE promoter is an inducible promoter as we discussed in previous class. So when this promoter is used, so now when we add estrogen, then only the mRNA will be produced and the protein will be expressed. So we used estrogen, then what will be the inference after adding estrogen? So the protein will be expressed in companion cells only, the protein will be expressed in sieve elements only, and sorry, the protein will be present in both companion cells and sieve elements, or the estrogen will not be help. The protein will not be expressed as estrogen is metabolized by the plant cells. So the correct answer is option C. And this is what I explained just now. So the companion cells and sieve elements are in constant connection because of the plasmodesmata. So they are in constant connection because of the plasmodesmata. Hmm, Sorry. So, and all the things of the sieve elements are controlled by the companion cells. So, if we express a protein within the companion cells, will also be present in the sieve elements also. So it will be present both in companion cells as well as in sieve elements when estrogen will be used to express the protein. Okay. So now we'll move on to the next question. So you are expressing beta-1,3 glucanase under a DR5 promoter in the xylem pole pericycle cells. What can you expect in terms of aniline blue staining and lateral root density? So as we discussed, that DR5 promoter is used as an oxygen response promoter. Only it will function only when oxygen will be present, and beta-1,3 glucanase is the callose degrading enzyme. So they are expressed in a xylem pole pericycle cell that helps in the formation of lateral roots. So what can you expect in terms of aniline blue staining? Aniline blue staining is basically used to detect callose. So whenever it will give blue color when callose is present there. And we have to find that what is effect on the aniline blue staining and lateral root density. So what will happen here is when beta-1,3 glucanase is expressed, so callose synthesis will decrease, and as callose synthesis will decrease, so there will be low root density there. Okay, because as we have seen that there will be high root density when callose will be present. So so the correct answer would be that less blue color will be seen and low root density will be observed. Okay, so here is the explanation that I was talking about. So as we can see that callose deposition helps in increase in root density of the plant. So here they used different mutants that have mutated beta-1,3 glucanase. So when the last one, two, there were mutants, so they showed increase in root density as compared to the wild type. Okay, and as we know that oxygen levels first increase in the pole pericycle cells, and that leads up to the tip of the lateral root. So when we express a protein under DR5 promoter, that is beta 1,3 glucanase, so here oxygen levels will be high. So expression of beta 1,3 glucanase will also be high, and that will help in less formation of callose rather than degradation of callose. So this will help in formation of less intense blue color and hence low root density. Okay. Now moving on to the next question. So what are the different methods used for transforming plant tissues? So either we can use electroporation, a gene gun, agrobacterium mediated, or all of the above. So yes, the answer is all of the above. And here is the explanation. So first is the biolistic method or the gene gun method. So what we use is we use a tungsten or gold nanoparticles coated with the DNA that we have to use that we use to transform on the plant, and using a very high speed and in vacuum chamber, we bombard it to the plant cells or the callus, and they are inserted into the cells. Okay, so this is the gene gun. And electroporation, sonoporation, or optoporation, in this case, what we do is we try to create pores in the plant cell wall, and and after that, the DNA enters into the cell. And this is the cationic transfection. So we bind the DNA. DNA is a negatively charged molecule with a positively charged molecule. And as we know, the cells have a negative charge. So if we can, like the combination of positive and negative charges will attract each other, and followed by the combination of electroporation or heat shock method will create pores in the plant cells, and the DNA will enter into the cells. And in case of magnetofection, what we do is we use magnetic field to create the pores, and then incubation with the beads also helps in creation of pores, and that will help in entering the DNA into the cell wall. And then micro injection. So we use a capillary needle or an injection, and using this we inject the DNA that needs to be inserted into the plant genome indirectly into the nucleus by holding a single cell under the microscope. Okay, and here the third option, agrobacterium mediated, is a biological method using which we use a bacteria called the agrobacterium, and we transform the cells by its infection. Okay, so these are the different methods that are widely used in plant biotechnology for generation of transgenic plants. Okay, so let's move on to the next question. So what is the use of acetosyringone in agrobacterium mediated infection to plant? So it helps in the formation of infection tube. It is a chemical attractant. It helps in division of bacteria. It helps in growth of plant. So the correct answer is it acts as a chemical attractant. So this is the procedure that we use for developing a transgenic plant using agrobacterium. So what we do is we take seeds or callus, and then we sterilize these, and we grow in a medium and a callus generation medium. So this is the callus. So now what we do is simultaneously we grow agrobacterium in culture, and then we infect the uh, cells with the agrobacterium using co-cultivation method, and here we apply acetosyringone. So it's a chemical attractant that attracts the bacteria to the plant, and in the normal scenario what happens is this is a dicot plant, and when this dicot plant is injured, so it releases some phenolics that attract the bacteria. Okay, and in this case, you can see there is no injury done, but as the callus tissue is very soft tissue, so the bacteria can easily invade the cells. And after that, we grow on a selection medium and using different antibiotics. So here the calluses that have the gene will grow on that particular antibiotic because of that antibiotic resistant selectable marker used in plant, and the seeds which the calluses which do not have this antibiotic resistant gene will die out in this medium itself. And then we shifted to shoot and root regeneration medium, followed by hardening and the growth of the plant. So this is the full process how we grow transgenic plant using Agrobacterium tumefaciens. Okay, so this was the full process. So moving on to the next question. So what must be the root inducing medium contain in a plant tissue culture medium? So it must contain high oxygen amount or high cytokinin amount or high amount of low oxygen plus low amount of cytokinin, or the last option, low amount of oxygen plus high amount of cytokinin. So it must have high amount of oxygen and low amount of cytokinin. So here is the things that we generally use in the plant tissue culture. So if we use in a normal nutrient agar medium without any hormones, then it will have no growth. But if the ratio of oxygen and cytokinin is kept same, that is, then the callus is generated. But if we have to generate roots, then amount of oxygen will be high and the amount of cytokinin will be low. And if we have to generate shoots, then the amount of oxygen will be low and the amount of cytokinin will be high. So here in the growth in the generation of callus, we must know that we have to use a oxygen to cytokinin ratio. Oxygen and cytokinin must not be too high or too low. Okay, and here we generally use indole acetic acid in the laboratory as oxygen and kinetic for cytokine. Okay, so here is the combination of oxygen to cytokinin that we normally use in the plant cell culture. So moving on to the next question. Hygromycin is used in tissue culture because it induces inhibition of protein synthesis, it induces oxidative stress in plants, it is used as a selective biomarker for transformed cells, or it is used in agrobacterium infection. So hygromycin is an antibiotic that induces inhibition of protein synthesis and oxidative stress in plant. So it is generally used as a selectable marker when we use a recombinant DNA. So we use a selectable marker HPT gene, that is hygromycin phosphotransferase. So that will directly inhibit or phosphorylate the hygromycin that will be non-functional in plant. So the plant having that gene will help the plant to survive in the to survive in so when the plant will have hygromycin resistant gene, so that will help the plant to grow on the medium that is containing hygromycin, and that is used to select the transformed versus the non-transformed plant. So here the answer would be A and B both, because it helps in doing this to the plant, and then also the plant is selected. Okay, and here is the full plasmid of the agrobacterium that is used, and this is the recombinant plasmid. So this is the disarmed tDNA region. So this is the left border, this is the right border, and after that we use a selectable marker gene, that is a hygromycin resistance gene that helps in growing under the influence of the antibiotic. Then the promoter of this gene to express this protein, and then the promoter of the gene of interest that we needs to be integrated into the plant. Okay, so this is the general structure of the disarmed tDNA region that is used in the recombinant DNA technology of the plant cells. So this comes to the end of our session and the discussion of week 4 of plant development biology course. Hope if you like the discussion videos, and thank you for your support.